I wanted to start the talk just by putting inherited platelet disorders into context. And obviously here is some data from the UK HCDO registry about bleeding disorders. As we all know, we've got about 27,000 people in the UK registered with heritable bleeding disorders, of which just over 2,000 are registered with inherited platelet disorders. So that's about 1 in 30,000 people in a population of 60 million. And we can see already that that's comparable with disorders like haemophilia B or factor XI deficiency, which have a much higher profile. And actually that's almost certainly a significant underestimate of the true prevalence, particularly of mild inherited platelet disorders, which don't tend to get registered in a national registry. The other important difference here between inherited platelet disorders and the rest of these coagulation factor deficiencies is that almost all of these are simple monogenic disorders. Inherited platelet disorders are, of course, highly heterogeneous genetically. And that's illustrated very nicely on this figure from a review last year, actually in Blood, which lists the genes implicated in human platelet disorders as described at the end of 2016. And we can see there are more than 50 already. And these are subdivided very nicely here. We have genes encoding platelet receptors, which manifest as platelet function disorders, genes important for the biosynthesis of some platelet organelles, notably granules, again manifesting as platelet function disorders. But lots of other genes here, growth factors, transcription factors, and importantly cytoskeletal regulators that are all important for the production of platelets. Defects in these genes manifest as inherited thrombocytopenias. And of course we've got some crossover disorders, genes in this region here, Berners-Soulier syndrome, where there's both defects in platelet number and platelet function. So the message really from this slide is that genetic platelet disorders are highly heterogeneous. Why then should we try and resolve the causal genetic defect in our patients with inherited platelet disorders? Well, I want to illustrate this with a case from the Bristol Clinic actually. It's actually two families, very similar structure and very similar clinical presentation. In both cases, the index case here is a female patient presenting in first pregnancy with unexplained thrombocytopenia at booking visit. In both of the families I'm presenting here, this finding isn't unusual per se, but what is unusual is that our index case has got a first degree relative and a second degree relative also with long-standing stable thrombocytopenia. This combination suggests a monogenic disorder, it suggests heritable thrombocytopenia, and it suggests an autosomal dominant platelet disorder. Here's the best available phenotype test we've got at the moment for investigating mild non-syndromic thrombocytopenia. It's a blood film. Our first case shows mild thrombocytopenia with normal sized platelets. Our second case shows, well, mild thrombocytopenia with normal sized platelets. It's the same blood film. We can't resolve diagnosis any further with phenotype tests in our laboratory. If we proceed to genetic diagnosis, our first family's got a causal variant in the CYCS gene. So that's cytochrome C gene causing what's formerly known as thrombocytopenia 4, a mild benign thrombocytopenia that's non-syndromic and associated with no bleeding symptoms. Our second indistinguishable family otherwise, in fact, has got a causal variant in RUNX1. That's quite a different thrombocytopenia disorder that's frequently associated with additional platelet function defects, notably storage pool disease, and also, for some variants at least, a 50% chance lifetime of developing myeloid malignancy. So that's familial thrombocytopenia with a propensity to acute myeloid leukemia. So quite clearly here, genetic diagnosis has allowed us to provide very different estimates of prognosis for our two different patients presenting with identical laboratory phenotypes in the clinic. And I don't need to convince the audience here, I don't think, that there are lots of other reasons why we should be striving for precision diagnosis in platelet function disorders in the same way we do for coagulation factor defects. What I'd like to do in this presentation is just review the current approaches we have available for genetic diagnosis in platelet disorders. So I'll be starting with candidate gene approaches, talking something about gene panels, but actually focusing on advances, what's happening now and in the future. So I'll spend a bit of time towards the end talking about gene discovery using whole genome sequencing, and then finishing up is where I think we're going next, in diagnosis of disorders, what techniques we need that are emerging now. So let's go back to the clinic then, just to mention in passing really candidate gene approaches. This is what we've been doing for 20 years in hemostasis. Here's one of my patients who you can see is presented with some mild mucocutaneous bleeding early in life. I think like most centres we would measure some coagulation factor levels, but also move very quickly to study platelet glycoprotein expression by flow cytometry. Here's his result. Here we've measured platelet surface expression of CD61, so that's the beta-3 integrin, and we can see that compared to a healthy control, our patient's showing markedly diminished, nearly absent expression of this platelet glycoprotein. So this corresponds obviously to the well-known and well-characterized platelet disorder Glanzmann's thrombocythemia, in which we all know you're deficient in either the beta-3 integrin or its partner, the alpha-2b integrin. Deficiency of two proteins indicates two possible candidate genes to look for causal variants. Here's one of them, the ITGB3 gene. So if we want to offer genetic diagnosis in this family with this distinctive phenotype, we've got a manageable task on our hands. We can tackle each one of these coding exons in this gene individually by PCR analysis. We can then subject each of those PCR amplicons to a tried and tested sequencing technology. This is chain termination or Sanger sequencing, and look within that for sequence variants by comparing the sequence to a human reference sequence. And in this example, there's one variant here and a second variant in another exon in this gene, consistent with recessive inheritance. Finally, we compare our observed variants from direct cycle sequencing to very well-studied and well-curated disease-specific mutation databases, and one exists here for Glanzmann's thrombocythemia, or alternatively to larger variant databases like ClinVar or HGMD. And if we find the variant observed in our patient is also present in other unrelated families with the same disease phenotype, hey presto, we've got genetic diagnosis. So what this fairly straightforward example in Glanzmann's thrombocythemia is telling us is that because we've got a very specific phenotype test, flow cytometry, we can narrow down the search for candidate genes to one or two, amenable to tried and tested individual sequencing through Sanger technology. And because it's a well-characterized disease, no problems with interpreting the variants because there's a large repository of information about previously described families. So far, so good. Well, how about this, a different phenotype? Here, there's also a bleeding phenotype. The clinical phenotype here is mild mucocutaneous bleeding, some surgical bleeding. Here, the laboratory test is a platelet function test. It's light transition aggregation. And we can see immediately here that our patient P1 is showing diminished aggregation responses to the ADP agonist, a right shift in the dose response curve, but results that are similar to healthy controls with these other receptor agonists, the PAR1 receptor agonist and the GP6 receptor agonist. So what we know about platelet biology is that if we stimulate platelets with ADP, we achieve aggregation in the light transmission test by ADP interacting with just two receptors on the platelet surface. So it's fairly tempting to say, well, there are two candidate genes for this disorder as well. It's either going to be P2I12 deficiency or possibly P2I1 deficiency. But of course, in this example, that's a simplification because we fill in this arrow. It's not just a simple interaction. In fact, it's a much more complicated pathway. And in fact, it's pretty obvious to see here that if we have patients with diminished aggregation responses to ADP, then we have to think of lots and lots of different potential protein mediators and lots of different potential genes. So this is not amenable to simple tailor-made Sanger sequencing. We have to adopt an approach that allows us to simultaneously examine the sequence of lots of different candidate genes. Unsurprisingly, we now have a methodology to do this. This is next generation sequencing. It's been with us for about a decade. And I've just summarized on this slide one of the available technologies just in simple form. The starting material, just like Sanger sequencing, is genomic DNA, normally obtained from peripheral blood samples. First step in our process is to fragment the DNA by sonication. And then the most important step, we mix our patient's DNA samples with a library of baits. Baits are usually mRNA sequences, typically 200 mers, that bind to the specific target regions we want to amplify in our target DNA sample. Each bait's got an adapter on the end to allow us to manipulate it. After ligation, we immobilize our bait DNA fragments onto usually a solid membrane, where they undergo a PCR amplification step mediated by the adapters here, and then simultaneous sequencing, all at the same time, everything that we've captured. The kind of outputs we get look like this. Here's a genomic sequence, and our baits here are crucial. We've designed them to be overlapping and to cover our genetic area of interest. And when these are lined up by software, you can see that each individual gene is covered by multiple baits. We have multiple reads. And by lining again this with a reference sequence automatically, it's very, very easy to spot here the blue and the red lines where there are mismatches between our lineup and the reference sequence, and these are potentially causal variants. So crucial to the process is the design of the baits, and we've got several different options here. If we want to target a specific group of genes, then we select coding sequence of just those genes of interest. So that's a gene panel. We may want to take an off-the-shelf package where someone else has designed baits that cover all the coding exons. That's whole exome. And we can even now off-the-shelf take a package that covers not just the genes, the coding genes, but also all the other stuff. That's whole genome sequencing. And actually all three of these approaches are now in operation in the NHS as diagnostic tools for different rare disorders. But I'll start just by describing gene panels, because this is the tool that's available to us right now diagnostically for coagulation and thrombotic disorders. So this is the second part, gene panels. There are several of these in operation internationally, but I've chosen the thrombogenomics panel. So the thrombogenomics panel is a gene array, 78 different genes that have been previously linked unambiguously to human hemostatic disorders. It targets the coding region of those genes and selected regulatory elements, notably some promoters and some splice regions in introns. And if you look at some of these genes, you can see that what we've got covered here on this panel are all of the coagulation factors. A number of genes responsible for platelet function disorders and platelet number disorders, but also actually some thrombotic disorders. Here's how the pathway works in the NHS. Just like conventional Sanger sequencing, patients are invited to give consent and a sample of usually venous blood from which we can extract genomic DNA. Whatever the clinical phenotype of the patient, that genomic DNA sample goes through the same thrombogenomics panel. All 80 genes are studied. There's a bioinformatics step. Results are discussed and interpreted by an MDT meeting, and then passed back to the referring clinician in a formal report in the way that we're all used to receiving reports from genetics laboratories. The most important step on this slide is not actually the sequencing technology. That's reasonably straightforward. It's standardized and well quality controlled. It's the MDT meeting, and here's the reason. Here's the output from just one patient. Here's a patient again presenting with autosomal dominant mild thrombocytopenia, actually in this case not associated with bleeding. Now that we're studying nearly 80 genes, we're having to deal with more variants. And in fact, in each of these blocks representing genes, an M symbol represents the presence of a variant, a difference to our reference genome. And we can see that that's present in more than 85%. So we've got to deal with 50 different variants before we even start just in one patient with this panel. Now, many of those can be filtered out early bioinformatically by arguing that, well, if they're common in population databases, they're unlikely to be related to a rare bleeding disorder, so they can be excluded. If they're in the coding sequence and they don't change the amino acid structure of the protein, then again, they're unlikely to be causal, so they can go. But that still leaves us with variants that are potentially causal. And in this individual, we've got 13 different variants, all potential candidates, all present in heterozygous form. This is where the MDT comes in. And I just want to illustrate how they would deal with it just by two of these genes here, DTNBP1 and ankyrin 26. This is the kind of output the thrombogenomics MDT meeting deal with. Perhaps we look at the top row first. This refers to this particular variant in DTNBP1. What the software is doing here is annotating this variant against the major or official, normally CCDS annotated transcript. So the real transcript that gene encodes, here given its NCBI accession number. And by annotation, we mean it's simply saying what's the impact of that gene change on the gene on that transcript. What does it do? Is it coding sequence? Is it an intron? And we can see immediately here that that's placed this genetic variant at DNA level well deep within an intron in this gene. So we can say immediately that's unlikely to be causal. It's unlikely to confer loss of function to that gene. Not so with this second one. Actually, this places it as in a splice site. And we all know that splice site variants can potentially affect expression of genes. But here the MDT comes in. Two reasons why this is unlikely to be the causal variant. Well, this gene is the causal gene for Hermansky-Pudlak 7. So we expect people to have a platelet storage pool disorder and a pigment disorder. This patient's got mild non-syndromic thrombocytopenia, so it doesn't explain the clinical phenotype. Moreover, Hermansky-Pudlak 7 is a recessive disorder. This variant is only present in heterozygous form. So the MDT meeting will then take this as proof that this variant is unlikely to explain the disease phenotype observed in the patient, and that's why we need a clinical MDT meeting. Contrast that with the other example gene change here in the patient. Here, ankyrin 26, this particular variant at DNA level is present in the untranslated region of the major transcript for this protein. So superficially, you'd think unlikely to be disease causing, but actually, that region is the main promoter region. It's the mice and fly1 and runx1 binding site of the ankyrin 26 gene, and single nucleotide variants in this region have been previously associated with mild autosomal dominant thrombocytopenia in now about 20 or 30 pedigrees worldwide. So there's previous history of this variant causing thrombocytopenia. That's picked up the MDT meeting, and actually that's reported as the causal variant. If we look at the performance of panels like the thrombogenomics panel, we can see that if we examine cases where referring clinicians either know the causal variant already by Sanger sequencing, or they've deployed phenotype tests like coagulation factor assays, where there's a very good pointer towards a specific genetic disorder, next-gen sequencing panels like this give us the answer all the time, or most of the time. Not quite so good for samples that have been analysed where doctors can't give a phenotypic clue as to where the disorder is likely to lie. Only 10% of these patients got the answer through thrombogenomics panels. Now, of course, this doesn't indicate a problem with the technology. That works very, very well. What this is actually telling us is that collectively, if we do phenotype tests in our laboratories on patients with bleeding disorders, if a test doesn't give us a strong clue as to where the disorder is, actually it's not going to lie in the gene that we already know about. It's going to be somewhere else. And it's that somewhere else we have to look if we want to achieve diagnosis in this group of patients. And that other place is, of course, the rest of the genome. So the third section of the talk then, gene discovery in the whole genome. We've known lots about the architecture of the genome. Actually, for 17 years, we've now had the results of the Human Genome Project. And that told us very early that there were 3 billion bases. Lots. In the most recent build from Ensemble of the human genome, it's now estimated that there are only 21,000 different coding genes. There's 21,000 capable collectively of generating nearly 170,000 different protein transcripts. And if we aggregate all of the variants that have so far been described in human sequencing projects through initiatives like the UK 10,000 Genomes Project, to date, 63 million different variants have been identified. And that number is escalating rapidly. Again, if you look at data from UK 10,000 and also the American Exome Sequencing Project that surveys and analyzes these data sets, one individual, even just within the coding region of genes, has got a total of 14,000 on average single nucleotide variants. We add to that indel variants that aren't it, so even more. Of course, not all of those are going to be conferring loss of function to the genes, but many are. Incredibly, each one of us has got an average of 100 loss of function variants, so that's something guaranteed to affect expression of the gene, 100 loss of function genotypes. Fortunately, almost all in heterozygous form or in proteins that aren't disease causing. If we're doing whole genome sequence on patients with platelet disorders or other rare disorders, the challenge is pretty obviously then resolving the 1 or 2 causal disease variants from this blizzard of incidental bystander variants, some of which have compelling arguments for the loss of function. So what I'd like to convince everybody is that you need 2 separate things to make this happen. And the first is obviously statistical power. Whole genome sequencing is not a diagnostic test that can be done in isolation in individual patients or even families. It has to be done in the context of a large consortium where statisticians compare whole genome sequence data with lots of other people. There are several international consortia. Here's the biggest one. It's the Bridge BPD study, now subsumed into the UK NIHR Bioresource Rare Disorders, soon to be subsumed further into the UK 100,000 Genomes project. At this stage, this was a consortium of around about 40 international centres, and there are many contributors to this initiative here in the audience. And it's truly international, in fact, truly intercontinental. At this point, it had enrolled nearly 1,200 actually, individual index cases with uncharacterised, so no obvious causal phenotype, bleeding or platelet disorders. And for each one of those people, whole exome sequencing, or for the majority, whole genome sequencing was performed. So we need power. We also need a systematic way of describing our patient's phenotype. And the nomenclature that's used for the bioresource and also for the 100,000 Genomes project is an ontology system called human phenome ontology. And this takes a bit of getting used to. It's a systematic way of describing the clinical and laboratory characteristics of our patients in coded form, so with fixed semantic terms arranged in a hierarchy from general terms at the top to specific terms at the bottom. You can generate from these terms specific node diagrams, and here's a node diagram of someone with grey platelet syndrome actually. And if you can read at the bottom, there are 2 phenotypes here in the bottom row. Number of alpha granules is abnormal. Alpha granule distribution is abnormal. They are in turn a defect of alpha granules, which is in turn a defect of platelet morphology, which is in turn a defect of blood and blood-forming tissues. They've got a big spleen. So they've got splenomegaly, which is a disorder of a spleen, which is a disorder of an abdominal organ. So you can see this works in individual phenotypes that are expressed as strings of terms. The reason that's important is that you can mathematically express or describe complex disease phenotypes to enable statistical comparison between unrelated families. And that's important for the statistical genetics folk to be able to resolve causal variants in whole genome sequence data. Here's an example of how it's worked. Here's another patient from the Bristol clinic actually. Our index case here was investigated for mild bleeding symptoms that were probably clinically insignificant, but what she did have was a blood phenotype, reduced platelet number, increased platelet size, mild neutropenia, and in addition, hearing loss. Hearing loss that developed in the first decade of life. So that's pretty unusual, but what was even more unusual, it's got 4 children, and 2 of those had exactly the same combination of phenotype, macrothrombocytopenia and hearing loss. 2 had neither. So again, this is a pedigree showing us there's a syndromic disorder here that's probably monogenic and is probably herited as an autosomal dominant trait. Now I know what many of you are thinking, this has got to be MYH9 related disorder, hearing loss, macrothrombocytopenia. And of course that was considered first as the candidate gene, and this family went through the thrombogenomics panel. But MYH9 was not the causal gene, there were no sequence variants in the gene. So therefore, the pedigree was linked into the Bridge BPD study and underwent whole genome sequencing. Here is our patient's phenotype expressed in HPO terms. Here's the node map, and we can see we've got nodes here for thrombocytopenia and sensorineural hearing loss. And this pattern was used mathematically by the statisticians after whole genome sequencing to look for similar families in the cohort with the same nodal pattern. The principle of the analysis here is that if you've got two unrelated families within a large cohort with mathematically similar complex phenotypes like this, then if they've got a variant in the same gene, well, that's likely to be the causal variant, particularly if that variant's not present in other families. The statisticians can actually estimate that very precisely, and there are several different statistical approaches here. Here, the technique that's been employed is called similarity regression. And put simply, what they've done here is they've considered all the high impact variants in all the genes in all the patients in the bridge BPD cohort. So that's tens of thousands of high impact variants. And they then simply asked the question, what's the probability based on phenotype that two unrelated families with variant in the same gene are actually truly associated? And there are very, very, very few genes here with a very high probability of a true association, and one of which is this one. It's a gene called DIAF1. When we go back to look at the DIAF1 sequence in our index case, we find indeed a high impact variant. It's a chain truncating variant actually in this part of the protein towards the carboxyl terminus. When we go back to our families, we find that this variant is present in our index case. It's present in the two children with the same phenotypic association, but is absent from the two unaffected children, so there's segregation. What about the other family that we found? Well, there's just one other family with the same phenotype actually, a family that's unrelated. And once again, we get complete segregation of an identical variant in DIAF1 with these two major components of the phenotype. So what we've got after all this is actually statistical proof that this variant is the causal variant for macrothrombocytopenia and hearing loss in this family, a completely new platelet gene. Is that enough? Well, actually, it's not enough. We have to answer some other questions. Is it actually going to be a biologically plausible gene? Well, it is actually. DIAF1 encodes a protein called diaphanous 1, which is important in the cytoskeleton. Here we can see it assembling actin monomer into actin filaments, and we know actin is important in platelet production from proplatelet extension. We know that DIAF1's already been implicated in the mechanism of proplatelet function. It's a negative regulator of proplatelets, and it's even been implicated in hair cell development. So yes, it is biologically plausible. What about experimental proof? Well, of course, we can test experimentally whether this is going to impact on platelet production. Here we've overexpressed the variant DIAF1 protein in some HEC cells and looked at cytoskeletal organization. Cells here that have taken up DIAF1 show abnormal cytoskeletal organization compared to those cells which haven't taken up the DIAF1 transcript. So we can show it impacts on the cytoskeleton. In an animal model, this is work done by Sarah Westbury in my lab with Kathleen Fresson, where we've knocked down DIAF1 in zebrafish. And lo and behold, the knocked down zebrafish have both a hematological and a hearing, I should say balance, phenotype. So it helps confirm that this protein is implicated in hematopoiesis and in ears, even fish ears. Finally, the Japanese group here have targeted mice, a small animal model, by knocking in the exact variant observed in patients. And lo and behold, those mice are deaf. And if you study the inner ear by electron microscopy, they have reduced numbers and abnormal morphology of stereocilia. So experimental proof. Finally, can this finding be reproduced in other pedigrees? Well, actually, since discovery of this gene change through similarity regression, a very similar chain truncating variant being discovered in another UK family with an identical phenotype of hearing loss, neutropenia, and macrothrombocytopenia that's segregating with the variant in 3 generations. Also since discovery, there are now 3 separate reports in 5 unrelated families, Europe and Japan, of similar or identical variants, again linked to an identical phenotype. So this combination of multiple reports in unrelated families and experimental evidence and biological plausibility is now considered altogether by the ISCH SSC on genomics. And if it's been described in at least 3 unrelated families and there's supportive evidence, DIAF1 is now considered a tier 1 gene for thrombocytopenia and is now included in clinical diagnostic panels. So the DIAF1 story is a very nice example of how we can use whole genome sequencing and statistical genetics and experimental proof to identify new causal genes. And it's a productive approach. Within the Bridge cohort, this is one of nearly 10 new gene discoveries for platelet traits. But actually, in reality, there are many, many more families in the Bridge BPD core and in our clinics where whole genome sequencing focusing on the coding sequence hasn't given us an answer. Where next? Where are we going to find causal variants in our families? Well, one possibility is that we'd be looking in the wrong place. Only a tiny percentage of our genomes comprises coding genes. In fact, 98% of the 3 billion bases we all have is non-coding. Maybe we should be looking in the non-coding space for causal genetic variants in our unsolved platelet disorder patients. In particular, should we be looking in regulatory elements, proximal regulatory elements, promoters, transcription factor blinding sites that drive expression of platelet genes? Or should we even be looking further away in long-range regulatory elements, in things like enhancers, repressors, or suppressors? We've already got some precedent for this. There are 2 platelet disorders where we've already got variants in regulatory elements. So it's ankyrin 26-related thrombocytopenia. We've already mentioned that, but also thrombocytopenia absent radius syndrome. But there are undoubtedly more. The challenge is actually working out where these long-range regulatory elements are. I just want to finish with some new, very new publications or work that's giving us a clue about where we should be looking. And the first of these is GWAS. This is a very recently published genome-wide association study looking for genetic associations with full blood count traits. And this, as you can see, emerged in Cell at the end of last year. The scope of this study is extremely large. The researchers, primarily led by Cambridge, looked at full blood count traits in 173,000 healthy blood donors of European extraction. They looked at associations between genetic variants and 36 different full blood count traits, including some platelet traits. And they looked at the relationship between a total of nearly nearly 30 million directly observed or imputed variants, so common, less common, and rare variants with different numerical abnormalities. And what they found, that was a total of more than 2,500 different haplotypes associated with a change in a different full blood count parameter. But this is important. Less than 6% of those were predicted to alter protein sequence. So that means they must be causing the phenotype, causing a change in the hemoglobin level or the platelet count or the white cell count by influencing expression of critical regulatory genes. If we look at platelets, 570 haplotypes were linked statistically with a change in a platelet parameter, the platelet count, the mean platelet volume, the platelet crit, or the platelet distribution width. So the immediate inference from these data are that if we know which genes these GWAS hits regulate through common or less frequent variants, then these will be very good candidates to look for large, rare variants, very rare variants with large effect size, possibly causing more dramatic changes in platelet parameters, manifesting as heritable thrombocytopenia, so inherited platelet disorder. And in support of that, if we look and see where common genetic variants occur related to platelet count, we've got some familiar looking genes here. These genes have got common variants identified in GWAS, but also rare variants linked to rare heritable thrombocytopenia phenotypes. So this tells us we're on the right track. We can go one step further and actually look to see whether those GWAS hits lie in areas of known regulatory activity. And here's one approach. This is epigenetic mapping, where we simply scan the genome in a particular cell, in this case megakaryocytes, the cell that's relevant for platelets, to look for biochemical modifications of DNA that indicate a particular region has got regulatory function. And I've presented some simplified data here really in a region of chromosome 14 near a gene called actinin-1. Now, loss of function variants in the actinin-1 protein are a very common cause for non-syndromic macrothrombocytopenia. That's a human disorder described about 5 years ago. These two traces here show regions of the adjacent genome that undergo a particular kind of histone acetylation, the kind of histone acetylation you get in enhancers. And we can see there's a very prominent peak just upstream from the actinin-1 gene, in proximity to the actinin-1 gene, suggesting that this region here has got a key regulatory element on this gene that we already know impacts on platelet number. Can we test it experimentally? Well, yes, we can. Again, here are new data, again from Cambridge, actually. These are ChIP-seq data. This is not yet published. ChIP-seq is a technique where you can look at DNA-DNA interactions. So in particular, the DNA-DNA interactions between an enhancer and the promoter related to a gene. And this circular view is a bit of chromosome 14 that contains, in green here, the actinin-1 gene we've just been looking at. And what these purple arcs indicate are experimentally confirmed interactions between DNA in megakaryocytes. And you get a different pattern depending on what cell you're looking at. And this is a megakaryocyte-specific DNA-DNA interaction. And we can see very nicely that there's lots of interactions here between the enhancer element that we identified in the epigenetic mapping and the actinin-1 promoter. Again, further emphasizing this is a key regulatory element. And it won't come as any surprise, it's the GWAS hit that was associated with reduced platelet count in the large GWAS study. No surprises, it lay within this enhancer element. So there's cumulative evidence here of specific regions within the genome being able to regulate important platelet number genes. This is a very recent analysis performed by Ernest Trureau, a statistician at Cambridge, who has actually used these multiple approaches to map the entire non-coding space. And what he found is there are several regions, actually small regions, that come up with lots of different hits of evidence for being key regulators of platelets. You get a blue square in this region here if you've got a GWAS hit for a platelet trait. A blue square in this region here if the SIP-seq experiments show that you've got a transcription factor binding that we know is important in megakaryocytes. And we've got a blue square here if epigenetic mapping indicates that region is an enhancer or a promoter. And we can see there are some regions here that are virtually all blue. There is overwhelming evidence that they are critical master control regions for regulation of some platelet genes. And in fact, they're very small regions, just 2.5 kilobases of the 3 billion genome that scores almost all blue hits. So what I'd like to suggest is that when we've given up or not found anything in the coding region of our platelets genes, it's critical regions like this we should be looking for novel genetic variants accounting for traits like thrombocytopenia in our families. Just to summarize, I've convinced everyone, I hope, the clinical importance of precision diagnosis by genetics. I'd like to leave you with a message that whole genome sequencing is already a proven tool for genetic diagnosis, for gene discovery. But you need large study cohorts, you need systematic phenotyping, you need statistical genetics, and I think you need to look in the non-coding space. Please just remember that whole genome sequencing has already entered clinical practice as a primary diagnostic tool. In particular, the UK 100,000 Genomes Project has been recruiting and doing whole genome sequencing for more than a year in most English centres. I'll finish and take questions.